Hey everyone, today's video is looking at how scientists can identify and also come up with different treatments for different pathogens that are infecting our body. So this specifically relates to the dot point you're seeing on your screen here, where we're looking for different strategies to help us identify and control the spread of pathogens. So there will be specific identification techniques that you need to be aware of, as well as the different role of antibiotics and antivirals in treating pathogen infections. Okay, now we're going to have a look at some techniques that scientists can use to help identify the pathogen that is causing a particular disease. Scientists do need to be able to determine whether it is a bacteria or a virus causing a disease, and then from there they need to narrow it down to a specific type of virus or a specific type of bacteria so that they can take the best course of action in terms of providing a vaccination, maybe antibiotics, antiviral drugs, that sort of thing, to help either prevent the spread or treat people who already have contracted that disease. So if we're looking at identifying a virus, some of the different techniques are listed here on the slide. I'm just going to briefly touch on them. You don't need to memorize these or know them in any detail, but it's a good idea to be aware of the things that scientists would be looking for to determine what type of virus may have caused a particular disease to outbreak. So obviously there's physical identification, which is where the scientists will be looking for physical characteristics that they can see on the virus particle. And they usually do that using either X-ray crystallography or an electron microscope. Remember, virus particles are extremely small. There is absolutely no way that a scientist would be able to see a virus particle using a regular microscope. They need to be using something much more powerful. And using these techniques will give them a physical structure of what this virus would look like. Some examples are shown in the images there. Another way a scientist can identify a virus is using immunological identification. And this is where they take advantage of things like monoclonal antibodies, where they have a stock of antibodies already stored for a particular strain of a virus. They will alter that strain of antibody by adding an enzyme to it. They will obviously interact that antibody enzyme complex with the virus that they're looking at. And if the antibody is complementary to the virus, so the antibody is designed to target that virus, it will obviously attach to the virus, as you can see here. And using a series of different chemical reactions, they can actually cause this antibody with the enzyme attached to it to send out a signal that the scientists can detect. And that then gives the scientist a positive result saying, hey, this is the virus that we're looking at. You guys have studied this before, you know how to treat it moving forward, that sort of thing. Obviously, this sort of technique probably wouldn't work if this is a brand new virus that scientists had never encountered before, because they wouldn't have an antibody stored to recognize it. Some other techniques they can use are the molecular techniques. So we've spoken about these previously. So things like gene probes or DNA or RNA sequencing as well. Whoops, don't know what happened there. So they can be used to find the actual sequence of the virus, and then they can design different chemicals or vaccines to target those sequences. So if we're trying to identify a bacteria, same sort of things happening here. I guess the thing that probably is going to most differ is the physical identification. And that's usually because obviously bacteria are larger in size. So we can use regular microscopes to do that. We can also use gram staining. You guys might remember from when we looked at the topic of immunology, we learnt about gram positive and gram negative bacteria. So the ones with the 3 layers, the ones with the 2 layers surrounding their plasma membrane, that sort of thing. So by staining them, obviously gram-positive bacteria are going to come up violet or purple in colour. Gram-negative bacteria come up pink. And that's a special stain that scientists know to use when they're studying bacteria, and it will just interact with that peptidoglycan wall a little bit differently and produce those different colours. Same sort of thing in terms of the immunological identification, so using those antibodies with markers on them to interact with the bacteria and hopefully get a positive result. And the same with the molecular techniques, using things like gene probes and DNA sequencing. They can take it one step further when they're looking at bacteria. If the bacteria has a plasmid, they can also do DNA sequencing or fingerprinting of that plasmid to help identify the bacteria that they're looking at. And the last thing that we are going to discuss is how we treat the infection of a pathogen or a disease. Okay, so if we're talking about the treatment, that means that someone is probably going to be infected by this. Okay, so a vaccine wouldn't work. So to prevent infection, these are things that we should be doing prior to. So things like disinfectants or antiseptics to try and make sure that the surfaces of objects are clean and don't contain the pathogen. But once an individual has become infected with a pathogen, there are a couple of courses that we can take. So if it is a bacterial infection, so the pathogen is a bacteria, we can try and treat it with antibiotics. Antibiotics will only work for bacteria. If, on the other hand, the disease or the pathogen that has caused the infection is a virus, antivirals can be used to sometimes treat a viral infection, but they're not as common as antibiotics. So we'll just quickly talk about what they can do. So when we're looking at antibiotics, and I'm sure pretty much all of you have had antibiotics at least once in your life to treat some form of bacterial infection, most of the time you're given a broad spectrum antibiotic, which is an antibiotic that will be effective against most species of bacteria, but not all. It's only if you have a particularly nasty or resistant strain of bacteria that you may need to take a second course of antibiotics, and it's usually a narrow spectrum antibiotic where the doctor has actually identified what type of bacteria has infected you. And that's because these narrow spectrum antibiotics are usually only effective against one or two species of bacteria. So regardless of what type of antibiotic you're taking, this image here sort of outlines to you the different things that an antibiotic could do to disrupt or destroy the bacteria from continuing to infect your body. Most of the time it's stopping replication by inhibiting the cell wall growth. It might stop protein synthesis. It might inhibit important enzymes, or it could be interacting with the DNA in the bacteria to prevent that replicating as well. So it's not necessarily destroying the bacterial cells, it's stopping the replication so that your immune system can catch up, and then your second line of defense cells and your third line of defense cells can actually remove those bacterial cells from your body themselves. When we're talking about antivirals, so only for viruses, these are only effective when the viruses are located within the cell and are undergoing replication. So these virus particles need to be in the host cell and replicating their DNA or RNA using the host cell's machinery. Antivirals will not work against a virus if they're one of those free floating virus particles that haven't entered a cell yet. And essentially they do similar things to what antibiotics do, except they're targeting obviously the virus RNA or DNA to prevent it from being synthesized or produced in transcription or translation. So we're slowing down the replication of those virus particles.